Hello everyone, welcome to our channel. In this video, we are talking about media and types of culture media. First of all, cultivation of bacteria. The process of growing microorganisms in a culture or any suitable medium by taking bacteria from the infection site or any other site or environment, such as like our body, environment, examples chair, tables, etc., then grow them in artificial environment in the laboratory, such as petri plates, test tubes, etc. Now the question arises, why we need bacteria? The reason of cultivation of bacteria is that we need bacteria because of industrial application, pharmacological application, environmental application, and research application. Now, culture media. Culture media are also called growth media. You can understand the meaning of culture media by their name, that they are specific mixture of nutrients and other substances, as I have already discussed in my previous video, that support the growth of microorganisms such as bacteria, fungi, molds, yeast, etc. Now move towards colony. Colony, microscopically visible collection of millions of bacteria originated or created from a single bacterial cells. Colony are those collection of bacteria which we can see with our naked eye. As you can see in this picture, we can see them on petri plate. Now, types of media. Based on their consistency, there are three types of media: solid media, liquid media, and semi-solid media. These types are based on physical appearance, and based on their consistency. Number one is solid media. Solid media contains 3% agar. It is used for colony morphology, means appearance of colony, pigmentation, and hemolysis can be appreciated. Example is nutrient agar and blood agar. Sorry, not nutrient broth, nutrient agar and blood agar. Now, second one is liquid media. Liquid media contains no agar. It is used for inoculum preparation, which is used in the further processes, blood cultures, and for the isolation of pathogens from mixture. It includes nutrient broth. Now, third one is semi-solid media, which contains 0.5% agar, and their examples is motility medium. Now we move towards the based on types of media on the basis of their constituents. Number one is simple media, and the examples is nutrient broth and nutrient agar. It includes nutrient broth and nutrient agar. Nutrient broth consists of peptone, which is a protein, meat extract, and NaCl, sodium chloride. If we add 2% agar in nutrient broth, then it forms nutrient agar. Here is the nutrient broth in a test tube and nutrient agar in a petri plate, which is a solidified media. Next one is complex media. The example is blood agar. It includes media other than basal media or simple media. In this media, special nutrients are added. For example, sheep blood or other special nutrients are added to make it complex other than the basal media. Third one is synthetic or defined media. Example is peptone water. This media is especially prepared from pure chemical substances for research purpose, and the composition of every component in this media are very well known. Example, peptone water, which includes 1% peptone and 0.5% NaCl in water. This media is especially used for those research purposes. Now next one is spatial media. The example is blood agar and chocolate agar. This media includes substances like blood, serum, eggs, and these are added to the simple media. This media is used to grow bacteria that are demanding in their nutritional needs. Some bacteria require special nutrients for their growth, so this media provides the special nutrients. Next is enrichment media. The examples are selenite F broth, which is especially used for salmonella and shigella, and alkaline peptone water, which is used for vibrio cholera. It is a liquid media used to isolate the pathogen from a mixed culture. The media is mixed or incorporated with some inhibitory substances so that the unwanted organisms cannot grow and their growth is suppressed. For example, when we grow specific media, some unwanted microorganisms can also grow in their media. So, some inhibitory substances are those substances which inhibit the unwanted bacteria. Now, selective media. Examples are Meconic agar for gram-negative bacteria, TCBS for Vibrio cholera, potassium telluride media for diphtheria bacilli, and eosine methylene blue for gram-negative bacteria, which we call as EMB agar. In this media, some inhibitory substance is added to a solid media to inhibit the contaminating bacteria such as antibiotics, dyes, chemicals, and alteration of pH. In this media, those inhibitory substances are added which inhibit or suppress or control the contamination of bacteria. Now, indicator media. Example are blood agar and Meconic agar XLD media. This media contains an indicator which changes its color when a bacterium grows in them. Indicator means it is a chemical substance or a dye which indicates whether the given compound is acid or base by changing its color. Next one is differential media, which includes Meconic agar, CLED, which is cysteine, lactose, electrolyte deficient agar, and TCBS, which is thiosulfate citrate, bile salt sucrose agar. It is a type of media designed in such a way that some substances is incorporated in it, enabling it to distinguish between bacteria. This media helps to distinguish between lactose fermenter and non-lactose fermenter. While as you all know, some organisms are lactose fermenter and some are non-lactose fermenter. This media helps to distinguish which bacteria are lactose fermenter and which are non-lactose fermenter. Next one is transport media, which includes Stewart's media, buffered glycerol, saline, etc.